Okay, so you can take some time to update the package. So what I will do from now on is to start the program by click this shortcut in the apps. You should be able to find this after you install. And then from here, you just do check for update. And then when you see yes, the program should download and restart the interface automatically. Then you should be able to done. So you don't have to search for the command line, the code. But for now, from now on, you just do this. Your package should be updated. So if you have trouble to find these buttons, and let me know, I will go through the procedure again. So here I just update the program and the package itself. So I just do restart. And then the latest version is 25.4.5. So this is the latest version. Now you can check. If you don't have this orange button in the app, you can, from here, help, you can do the install this app. This just gave you a shortcut in that app panel here, so you don't have to type the command line anymore. So today on our agenda is we will first visit this page in documentation to study CCA. Then we will look at a paper, describe the method called Harmony. Harmony is a batch effects correction program designed for single cell data. Suppose you can move the cell from two different batches around to make them align to each other better. Then we will look at the MATLAB function to do this dynamic time warping using function dtw. Then I will show you how you can use Monaco inside the toolbox. So Monaco 3 is one of the most popular single-cell pseudo-time analysis tools. And then I will show you manual trajectory. Remember, I mentioned my solution is not use any tools. I just use fade plus drawing the curve. So that's my solution. At the end, if we still have time, I will show you a quick example how you should do this highly variable gene comparative analysis, and then downstream after you found those highly variable genes which changed between two conditions. So what's next? Their enricher analysis, you put the top genes in enricher and to find the function enrichment terms. Another tool is called Metascape, another gene enrichment analysis tool online. Okay, so let's start with CCA analysis. I would like to do first do a search. try to find language model's answer from whether Surrey package is using this. It's using CCA to do the batch effect correction, and I don't know exactly how. So that's in the R. If you have the CCA, the Surrey package, you can run CCA to align two objects. So two objects are the single cell matrices, single cell RAC matrix. Then it will do the alignment for you. Come back to MATLAB. Let's try look at the documentation of canal. So this is a function to do the CCA in MATLAB. So we are just going to use the example documentation provided to do this. So we are going to use, we need two inputs, two matrices, X and Y, and then we get two rotation matrices, A and B. These two matrices will rotate, A will rotate X, B will rotate Y, to allow them to show the maximized correlation for their embedded results. So to do this analysis, we need to prepare two matrices, X and Y, for the inputs. So here is how we construct this example data, X and Y. So what you can do is to highlight these two lines of code, a copy, and put in toolbox. So this should give you a data set. So this data is one matrix. We are going to separate this into two matrices. So there are 5 columns and 406 samples. The first column gives you the value called the displacement. The second column is horsepower, and the weight, acceleration, mpg. So this is just some description of those cars or vehicles. Then the next step is we are going to remove missing data. So if you look at this table, if you browse down, there are some of these samples, for example, sample 11. We don't have information for mpg, so this is an incomplete record. We have to remove this. Similarly, number 12, number 13, number 14, 15. So we need to identify those rows and get rid of them, because the analysis does not allow to have these missing values. Fortunately, majority of them has no missing data, so we just get rid of a few sample points. So how can we do this? We are going to use index and remove rows. So this is the index. So we try to find any rows contains at least one not a number. So basically you have NaN indicator is not a number. So when you have the some rows contains at least one not a number, so this will be an index which indicate whether this row has NaN or not. So if you run this, I paste here, you will get a list of index, the yes, no, so logic index. So if you look at this NaNS, this is one vector contains 0 indicator. There's no missing value in this row. But if you move down, look at number 11, there's 1. Number 12, 1 means true. So this row contains not a number. So we are going to remove those. So what you will do is you take the negative, basically 0 becomes 1, 1 becomes 0 using this Tudor, the symbol here, and then get rid of those rows and also extract the column 1 and 1 to 3 to put into X, and the 4 and 5 put into Y. So this is the final step to prepare these two inputs. Okay, now we have two matrices of a different size. So you can imagine these are the two sets of measurements. In the first one is X using 3 variables, and the second is Y and using 2 variables to describe the same set of 392 samples. So we can visualize this description to see whether there's some structures among the data. So if you look at the first sample, we have 3 columns. So we can use scatter plot. Let's make a new figure and use scatter plot 3. So you can have first column as your X, second column okay, so this will create a scatter plot in 3D to show you the location of each sample described with the 3 numbers. If you have a higher dimension, you probably want to use a PCA to compress using top 3 PC to do this. But our sample, the input X, just have 3 columns. It's very easy. We just show them all. Don't do any dimensionality reduction. So this is the scatter plot we get from the matrix X for 392 samples. Any questions so far? It's clear? So for Y, we can do the same thing, but in this case, Y is only has 2 columns. So you don't need a 3D, you need a 2D. So this is for another set. So this is a 2D for Y. Okay. So in this case, X, Y are two matrices describe the same sets of samples. So you can consider this as a single cell data analysis. You have same group of cells. You have single cell RNA-seq and single cell attack-seq measured for the same cell. So you have two matrices. You know exactly they are matched to matrices by the rows because you sample the same cells twice, but just use different ways to describe the cell. So this is a small example, toy example we are using. It's similar as what we are trying to do if you have a single cell RNA-seq plus single cell attack-seq. I don't know how many of you heard about this single cell attack-seq. Who? You did it, you did it. Okay, good. So this is measured gene expression. So we know we have a matrix. You have a cell, X matrix cell by genes. So this matrix get from this technique also for the same set of cells. So if you have that multimodal technique, you prepare your cells and allow gene expression to be measured. And also this is an open chromatin structure. Basically tells you which location in, for example, in chromosome 1 and position 365 to 392. So there's a region in the chromosome, and you have a peak in this region, and this information is also individual cell level. So you have this peak information which tells you the open chromatin level, whether if you are a place where in this region there's a transcriptional binding sites, they're the open, more open to be binded, and then you have a higher peak. So this is the epigenetics level information. So in this case, you can just borrow the same framework to align this domain information with this to rotate the cells. So come back to our example here. So we are going to use CCA to rotate this and rotate this to allow them to get the largest correlation between data points. So what you need to do is to copy this piece of code, and then you will get several outputs. The output number 1 is a 3x2 matrix. Remember, our X is a 3x3. It's a 392 by 3. So X has 3 variables. So we need 3 variables to a rotation matrix A. But this will truncate to make them into 2D, because the second matrix Y is only 2D. So matrix B is a 2 by 2, so this is a rotation matrix for Y. We can do rotation matrix is B times Y first, so you rotate it. Can you do this? I don't think so. You have to use B times Y to match the dimension. Okay. So so that's what you rotate. So let's see, this is called V3V, small v. Let's see why. B times Y. So I just try to match the dimension better. Yeah, so this is rotation for the Y. Let's rotate A. So A is 3 by, small u should be A x times A. Okay, so this is rotate x. And R, so this also written R. R will be the correlation after rotation of these two matched variables. Let's see if I do the V, the first column with the U first column. I try to reconstruct this very R correlation between these two after rotation. We rotate back. Yeah, so I try to see whether this will have the same value as this R, the first R. It's not the same, so I believe there's some of this centralization in the program here. So I didn't get the directly just rotate X, Y using rotation matrix, but a similar idea. So the idea is to found the two rotation matrix to rotate original data to allow them to align. So let's see. This is output for A, okay. Kind of smaller. Yeah, so we can, let's get this, copy the whole thing of here to see the results. So this piece of code just create a 2x2 scatter plot. To show you the correlation. So the first correlation is between this after rotation, the first dimension of these two, and after rotation you get a very high correlation for the first dimension results. So this indicate this is 0.87. Well, the second against the second projected position, U2 with V2 have a correlation 0.63. Well, these two are orthogonal to each other, so you don't expect to have a strong correlation between V1 and U2 or V2 with U1. So this is this part of the example. Okay, so this is about it, and this is explanation. And what I try to do, I try to use A matrix to rotate the input X to get a U. The number I get directly is very slightly different from the function computed. I don't know exactly how inside there, which step I missed. So I was trying to do this, something like this. So to do the U is, so I try to do this directly on the data to get the capital U, but the number is slightly different from each other. So I don't know which step makes this difference. So I believe this internally their centralization steps for the variables. But that's the idea. You get two rotation matrix, small a, small b, to rotate input x, y matrices to get this uv after rotation, to allow them to correlate it to each other. Yeah, so you need to do this normalization in order to get the same number. So that's the explanation why I didn't get it. So, but if you do this, so that's what I'm doing. I use directly without the centralization, but if you use centralized, you and hopefully this U and the big U are the same, okay? Any questions? I hope you know what I try to show you here, okay? Now this small u is the same as computed from the function big U. So before I start, I ask how the slew rate package is using CCA. So one thing I don't understand is in these applications to run CCA, those two samples are from different cells. If you have two matrices, for example, one matrix contains 300 cells, another have 500 cells. So this example is very different from what we showed this. It's very different from this as well, because this you have matched number of cells. I don't know in slew rate how they deal with this situation. If you have two matrices, they are from two different sides of the cells. There's no one-to-one cell match between them. They all matched for the same set of gene. That's fine. So I don't know how this function is implemented. I never use it. Okay. But maybe inside we should open this. I haven't had time to look at this very carefully. If you have unmatched samples, how you do this CCA? Maybe I should ask. Yeah, so this is a small question for myself, so we don't have time to solve in the classroom here. Okay, let's move to the second topic. We are discussing, will be this paper here. Let's just visit this paper and see. So this is a paper published in Nature Methods. In 2019, it has been cited 3,793. So this is a really high number. This article describes the method called harmony. So I think the problem they want to solve at that time is when the data set I see it with the different technologies. Then you have the discrepancy or bias of batch effects introduced from both biology and the technique perspective, and they try to use harmony to do this project cells into the shared embedding, which cell grouped by cell type rather than data specific conditions. I think the whole purpose for this is they try to embed the cells and try to push similar type of cells to each other, to the same group. Okay, so let's see how we can use this method in MATLAB here. So let's try to get the available code. So this is where the source code is in R, okay? So let's see, we need to have R prepared. Remember, you have installed R. So the R is here, RStudio. If you have RStudio, let me know you have this. So I have RStudio. I need to know where I installed. So I installed R in the programs R 4.3. So this. So make sure you have this RStudio, the environment. Otherwise, you may have to reinstall it from the ZenWorks. Let me know you have this R. We are going to install Harmony in R, and then send the input data from MATLAB to the R and read back the Harmony results. Do you have this RStudio installed? Good. So from.
It's the most current version.
Yeah, 4.4. It's good. Is it okay? Yeah. You have this. Okay. Yeah. Good. You have R, right? Okay. Yeah. So if you give it a try to see how install Harmony according to the documentation in the Harmony GitHub website. You have R. Okay, everybody got R. Is this R? Yeah?
Yeah.
Okay, good. Okay. I just make this font bigger so you can see. So let's visit this page, see Harmony GitHub. So according to this documentation, we need to do this. I just copy this and paste. So this is a command line you need to use. Okay, so this is it's done. So this is what we use to install Harmony, and then you can test whether you are installing this correctly. You can call this, and then it's done. So if nothing wrong, and then it should be okay. Everybody is fine? Okay? Yeah. Still working on it. It should be okay. Okay, now we can come back to MATLAB. You don't have to worry about R anymore. From here, we can just load example data. Processed. Then from the label, the cell group, we can label the batch ID, so we know this data contains cells from 8 weeks against 14 weeks. Clearly, they're separated. if you consider this as a batch effect, you want to move them, put them onto each other. That's a harmony we want to use to do the job. So in external, we look at the setup R environment, because we need to find where the R is installed. Yeah, so from looking at this, currently my MATLAB is pointed to the wrong version of R. So I have to, so if you can see this clearly, this is R 4.3.2. Well, the one I installed, Harmony, is in different versions. So there are multiple versions of R installed in my computer here, so I have to find the right one to use. So to set up this, I have to use, see, I want to use another version and then search for the C program inside R and find the right version, and in the bin then select this. Okay, so this is a series click to point to the right version of the R environment. Then I'm set. So now if I check my R environment, this is pointed to the right version I'm using. So this is the place. Okay, so I point to here. You got lost. You don't have that.
So for in here, it would be on the tools.
Yeah, you can. No, it's not from here. It's from within the MATLAB.
Okay.
The tool, yes. Yeah, then you can do this directly here. Yeah,
Oh, okay.
click. It's still old, so you use another one. You go to the C, my PC, C, users. Oh no, program. So do you have the program files? No, up, no, this one. Found R. Click here, double click inside. Double click a bin, double click this and open. Okay, here you go. Yeah, yeah, you don't know where it's located. So click this.
Yeah, that has already been set. Actually,
Good. Yeah.
but I don't know how.
Input data and example. Okay, then process. Yeah, you're ready. And then you click this label, the batch. Yeah. So now you can click external, then find Harmony somewhere. I don't know. Oh, I removed that. So, yeah. Oh, I removed that column. Yeah, but I show you how to do it. Yeah. Yeah, so that's the setup. You got the, okay. Yeah. Good. Yeah, it's okay. You still have question.
After the installation. So
Have you installed it? Have you installed this? Yeah, you're right. Mm-hmm.
So then I just run it on this.
Yeah, so you do this CG. And from here, you found setup. Click this, and you are done. Mm-hmm. Yeah, then you load the example. Okay. Mm-hmm. Okay, I think most of you have already done this step. So because I already removed the harmony from this menu, so we probably have to do some editing of source code to get it back. Okay, so let me show you how to do it. I should put it back for classroom, but in reality I found it's useless, so I removed from from the menu. So let's do this. You do edit and SCGA2. So this will allow you to visit the source code where we are going to find the place to add back the harmony, the manual item. So you have to move down to here to a place. Let me show you which row. Yeah, so you should be able to get to the row 283. So here we just need to add one line of code, so you can have that back. Let me find the harmony. Oh, Python version. I'm confused myself now. So we do have harmony, but it's in the Python version. So let's just come back to the, so we don't have to change anything. We are going to use the Python version of the harmony and use Python. Let's just still load this. So what, I was confused by harmony in Python or in R. So we do have a Python version harmony here. So forget about the R package. We are going to install Python and the harmony Python version. So first, let's see, you have conda. Let me know if you have Anaconda, this one installed. So that's a Python we need. Good. Good. Yeah, because we had installed this before, so I hope everybody has this. Then from here, we need to set up a Python environment. Okay, you click this and see whether you you have the right path point to this. That's a place where I installed mini conda here, and then under this you have a Python exe. So you need to find this file, and you need to know where you installed Python. This should be okay. So once you have this, what we can do first, just run Harmony directly. I hope the program can install it by itself. If it's not installed yet. So that's what I want to try first. So I will see, this is a batch, okay? And then you just run batch integration and continue. Use a working folder to save in case there are some of the intermediate steps. So the program should check whether you have that installed. So I'm using 3D. Yeah. So you see this, the cells start to move close to each other after you run Harmony. You can run that again. Just do further alignments. You can say no. So you see after 2 circles of running Harmony, the cells has been moved, and somehow the batch effect is gone, and that's what Harmony is doing. You can find a way to save the coordinates. I'll show you later, but that's the whole idea. I don't know that Harmony does nothing else. It not change expression level, no imputation. It just gave you some idea about what a cell should be if there's no batch effects. Okay? I don't know how many of you can get this. Do you have this function? Yeah, yeah. How to, no, the wrong button. You don't need this. You need this button here. ID. Yeah. You only have one sample. You extract just all.
Yeah, I think I extracted it when I was.
Yeah, you
I
do.
sometimes mix up the extraction with the labeling.
Yeah, so you don't extract. You just do file import data again. So just yes. Again, just from. If you have no batches, you don't have to do the batch effects.
Okay.
Yeah.
And then I ran Harmony through the.
Yeah.
And then.
Use this.
But then I did this part. I guess I didn't separate the batches, so didn't do it properly.
You can say no for now. Yeah, do that again. Then
You
you already did once, but you can run that again to
okay.
make them even closer to each other. Yeah, so it's working on your computer at least. Yeah, it's good. So yeah, I know this is a place. Maybe I will fix this later. So you want to save the change the coordinates somewhere in to replace. Yeah. Okay, it's running. Good. Good. Yeah. So that's using it. Okay. Good. I think most of the time this is to make the paper's reviewer happier because they want to see, okay, you made the correction of the batch effects. Okay, so we can take a 5 minutes break. Okay. Okay, so next function we will look at this DTW. Okay, so in MATLAB we can found the example from DTW for the documentation. So this is a process of two time series signals, and if you look at this input, rather than big X, big Y, now it's a small x, small y. So in MATLAB, so capital variables like big X indicate a matrix, while the lowercase normally indicate a vector or single number like a k. But this one, we need two inputs. Okay, so these examples. Two variable examples. So x, y is defined, and the one is longer than another. X has 1,000 variables, 1,000 values. Where y has 399. So if you plot x, you get a curve. If you plot y, you get another curve. Okay. So this function allows you to generate this aligned pictures. So all you need to do is just call this function, and also not require any output. And you get this original signals, these two curves, and then align the signals to minimize Euclidean distance afterwards. If you ask output D, then it will only compute this distance for you. If you don't ask anything, it will just generate this picture. So in this case, we change the frequency. Yeah, so this is an example, and then the follow-up example is to use complex numbers. So in this case, this last example is to looks like a 3D stuff. This is a time axis. Then you have real number, imaginary part of this complex number, and then get the align the signals. Okay, there are some other examples. So all you need to know, their functions exist. To do this, and when you have the problem need to solve in the real applications, you may realize, okay, there's a function I can borrow sometimes later. So I don't know when. Maybe it's in the pseudo-time trajectory analysis when you have two set of samples. You build one trajectory of the time points, and we want to align with other. So in that case, you have two curves. You want to align them, or to find a way to minimize the distance, to change the position. To anyway. Okay, so any other questions? Now we just follow our schedule, move to Monaco 3. Monaco 3 is our package, so we need to find a way to install it. Installation, let's see. Remember, this is R, so we need to have this. We installed Harmony, which is unnecessary because we use Harmony Python version. But now, to do this Monaco, we have to install R package. So let's do this. So you can just follow the instruction on this page, copy each block of this source code, put into R, and hopefully at the end everybody can install Monaco 3 in your system. Okay, the last step is to do this develop the tools and then install this. So in the middle of this installation procedures, there are tools installation, so you just need to wait, and the all essential packages. Yeah.
Does it say mine is still uploading? Is that what it's?
I think that somewhere, let's see, download is. Yeah, so you need, where did it come from? So, yeah, so you need a place where it's to install this. That's what happened on my screen. So
Okay.
I don't know where you're, if you found this.
Oh, I didn't do this.
It's not this one. Yes, shut this down. Do you have the window pop up for this?
I don't.
It should be this.
This one?
Yeah, you just select maybe. I don't know what's this. Yeah, maybe just. Say yes. So this will do the same thing over there. So yeah, it takes some time. Yeah, you have to do that again. Yeah, to this part again.
No, right?
Say yes. You have to say yes because. I think you have to click this.
No help.
I don't know. I never had that. Maybe try second options. Yeah, I have some trouble to install this. So what I'm doing, I just try I get the same as this. So the credential helper. I just try to run Monaco directly from the interface to see whether I can get some luck that program installed itself. Because I couldn't install Monaco from reading the installation instructions, follow the in R, so I just tried to run this here directly. No, I don't think we can do it. I have trouble to install the package, so on this machine, some machine I can, but not this machine, so we cannot do it. Yeah, this is still the error message which tells me I have some trouble to install this when I run the source code. So I cannot do anything on this machine now. So I think we should move on to our own solutions. So remember, the trajectory analysis, you have to believe the cells you are trying to rearrange their trajectories in there. Okay. In this sample, we have different. If you look at the annotation, we have different cell types. We probably have enough alpha cell to model. So you don't want to build a trajectory, you jump from alpha cell to beta cell because it doesn't make sense biologically. So we need to find a potential trajectory. I believe we have enough alpha cells. So also alpha cell, you have 2 different time points. So this is 8 weeks, this is 14 weeks. So we know probably we can model the transition from the early weeks to the later weeks. So what I can do is to extract alpha cell out. I will click this button. Click this button to select cell type. And then from alpha cells, click OK. And then click select. So this will create another window, only contains alpha cell. Okay, so this is the cells we will build a pseudo time for. So I can close this. So in here, if you look at annotation, there's only one cell type. But this cell type is from 2 batches. If you highlight a cell with batch ID, you will see this and that. We can do some kind of filtering to get rid of some genes which expression level is too low. So I remove 80 genes. Okay, so this is the data I prepared for trajectory analysis. So now the embedding is using t-SNE. As I said, t-SNE is not the right way to do it. So we need to change to embed this into a 2D fate. So I will click this embedding button here and click no, because I want to re-embed into a fate 2D. And I select this method. And let it to do. Okay, so this is a trajectory. This is a cell arrangement, I believe, captures the global distance between cells. So you can see one sample is still highly developed. And this maybe there's a trajectory. Some of the cell is still in the early stage. So now we can see, okay, maybe this is a starting point here. Okay, and then we move to this end. You can have another trajectory from this end to here. I would see we can try from this end to here. Okay, we need to clean this up because this is a branch. We need to delete those cells. We want to build one trajectory for the cells which follow this trajectory. We don't want to model this branch, so we need to delete them. If you decide to move from this end to here, then you can delete those cells. So for now, I just try select those and delete cells, delete selected. So this is a structure. Now, what you can do is to plot this trajectory by select the plots and the cell trajectory. You can try spline fit, see whether the program gave you this automatically. So this is not very good trajectory. It's too complicated for the algorithm to find the parse. And also the density involved. So this is not a good solution. So we try another algorithms. So this time we use a principal curve. This is not too bad, okay? So we, if we believe it's start from this end to that end, we can switch this and then to view select genes. Yeah, so some problems here, so I will fix that. So basically this produce the trajectory for you, follow this principal curve which goes through the majority of cells. So you can use this as a pseudo time if you need. And then once you have this, you should be able to find your pseudo time, this assigned to each cell as a variable here. So you see a different method. So in case either of this split fit or principal curve method does not do a good job, you can do the manual annotation. So I show you how to do this, the last option. So if you select manual, here you need to draw a curve. And after you draw a curve, you can save to a file and you can load it back. So now I start to draw a curve. We will select points along this cell populations. At the end, then we just click button to stop. So I say yes, I start to draw the curve manually. So I click this, click that, and just follow the trajectory. I believe the cells are moving. And you click, keep doing until the end. Then you think, okay, I can finish. Then you just click return. This will be your manual annotation of the. You can save this manual annotation somewhere. Yeah, there's some problem here, so I cannot show the gene change. But this is a manual annotation, and to draw trajectory for your pseudo time. So each cell then will be assigned as a time. Yeah, so it's too hard for you. Okay, good. Yeah. Return key. Yeah. Done. Yeah, that's all. Can do it? No. Yeah. Yeah. Not working. Yeah. No, it's okay. Got it. Mm-hmm. Yeah. Yeah. So just update your tools because you are using older version. Yeah. I don't know if you can do this help and check updates. Yeah. Mm-hmm. Yeah. Just wait and it should be. Yeah. Oh, you got it. Yeah. So once you build this curve and assume this is time 0, and then you move small steps until time 1, so there become a continuous variable. This number will assign to each of the cells. Now you can label the cells by manual pseudo time, by color. If you see, okay, a new figure. So yeah, you start to see this color and indicated by the pseudo time. Okay. So this become a variable. This variable you can use to do the regression. It's already there, so we can try. So for example, if you select this scatter plot to show the relationship, you can select this and then found we want to use this pseudo time and then correlated with some gene select save genes, do a transformation. So here I'm plotting out the scatter plot between your manual annotated pseudo time for the cells against gene expression. You see this is interesting, and this gene become lower and then higher again. This is the opposite direction, seems going up and then down. So those are just some random genes to show their expression along the pseudo time. Yeah, so I don't know, maybe this just mean nothing, this all artifacts, because our samples was selected or sampled mature cells, and also week 8 to 14, they're too long in the middle. So maybe there's no continuous state change. So what we detect here, and nothing special, just artifacts. But that's just idea to show you if you have a real development time and you have cells from different stage, you draw a pseudo time curve, and this tool will help you to find those genes which really correlated with the time. So you can do a feature selection. Here is a new function to allow you to use lasso to do this select genes. You can select Y. So this Y here is our principal curve pseudo time. Then you do the transformation for genes. Then there should be a lasso application. Yeah, maybe take some time. Yeah, so this is a lasso analysis to find the genes that, say, top 10 genes change expression with the pseudo time. So you can find from thousands of genes in which top 10 give you the best regression. Yeah, so those are the top genes found out. Okay, so this is another way to help you to find the top genes to explain your pseudo time. So those are the gene names. We can look at the scatter plot between pseudo time with this genes expression of those genes I selected, top 10 genes. Do an imputation. Yeah, so those are the genes selected by the algorithm. Okay, so this is a pseudo-time trajectory analysis. We are using an example, real data example, but maybe biologically not a realistic. So there's no link between alpha cell well developed in week 8 compared to they're just isolated group, but we, for demonstration purpose, we just draw a curve between them. That's not true because they are not intermediate steps, but it depends on your applications. In some well-defined cell population, you know it's from this population gradually you have a small step change and it becomes this population. You really can draw a curve and between them and then regress the curve values and then found the genes which may drive or follow this change. Okay. The last step is I will very quickly show you again what your homework will look like. So we can just use this as an example. We have same population that's required for the same cell type. Just 2 different batches, okay? And then we want to find variability difference in some of the genes. So we do, in analysis, you click differential variability, and then you select a working folder. And also, this is a mouse data. We want to clean make its gene number small and also more informative. Now you select 2 groups. These 2 groups are compare these 2 using split fit method. And that's your results. Basically, you save these genes. Okay, so those are the genes we found have a major change in. So this is number 1, this is number 2. Maybe we try SST. So this SST changed its variability pattern greatly in week 14. Okay, so you can see the increase of this variability. And then we can visit the next one. Yeah, so this also changed. This become lower because you have more variability in early stage, which in the later stage, this gene become a normal gene without any variability. So you have those lists, top list of genes. Depends on the cell type, the samples you compared, so you can start to interpret why this later stage for this particular gene have an increased variability. The average may be different, similar. So these two genes, these genes in two samples, they're all lowly expressed, but somehow in the orange, in this later stage, the variability increased. So you need some interpretation for those. Depends on what cell type you are talking about. Also the direction. Write a small report on that. You can do enricher analysis. You found the top 200 genes and then send to the website. So this website allows you to see those top 200 genes. They are enriched in terms of function of the cell, cell dice, and also ligand receptor activity. Okay, because if you check every gene individually, you don't have idea what they are doing as a group. But if you push this top 200 genes into the enricher, you got the terminologies, which the top 200 genes are involved in the apoptosis, and some of these communications of using ligand receptors. So it gave you the idea. And you can also see which genes are in this group. All right, so 5 more minutes for the question before we leave. Okay.
If you go to add your data into the MATLAB and it says like the index is too big for the array.
Oh, which one?
I just had the paper that I was trying to do with it's like a spinal cord injury paper, and I tried to enter one of the samples into MATLAB, and it just said the index was too big, like array-wise.
Oh, maybe it's memory issues.
Oh, okay.
I don't know.
Yeah, I can try a different, like.
Better computer, maybe.
Yeah.
Yeah.
Okay.
It could be. Okay. See you next week.